Hello everybody, we're going to pick apart some of the test questions for Biochem 2 exam number 1 from Fall 2013. Alright, so the first one, which of these is an enzyme redox cofactor? That's going to be FAD. It could also be FMN, NAD+, or any of the reduced versions like NADH. Those are all redox cofactors. This is found on Unit 1, Slide 51, titled B vitamins. Always used in dephosphorylation reactions. Remember, protein phosphatases are the ones that do dephosphorylations, like PP1, protein phosphatase 1. They remove a phosphate group by using water to do that. You can find that in Unit 3, Slide 72, where magnesium is used for your kinases. These are going to be used in redox reactions, but not all the time. These are only used in some redox reactions. Class of enzyme with 3 substrates. That's going to be ligases. Substrate, substrate plus ATP. So that's what he's talking about there. Number 4, partially activated by lipids. If you look through these, he's talking about PKC, which is activated partially by 2 DAGs. He also say which of these is partially activated by an inorganic molecule or a mineral. And then he'll be talking about 2 calcium. Just remember, PKC is partially activated by this or this, fully activated if you have both of them. That's in Unit 2, Slide 49. It illustrates that. Number 5, let's review. A is the answer. Activated carrier, transfer of CO2. If you look at the cofactors of metabolism slide, Unit 1, Slide 50, you'll know that biocytin transfers CO2. These are going to transfer electrons. TPP is going to transfer aldehydes. And CoA transfers acyl groups. So make sure to review that. It usually throws one of those on there. They're a little trickier. Which cell generates most of the energy they need from the complete oxidation of glucose? It's going to be your brain cells. Brain cells can use a little bit of ketones as well, but they're using mostly glucose. They can't use fatty acids. Red blood cells use only glucose, but it's incomplete oxidation. It only goes to lactate. And then these other 3 use a combination of glucose, fatty acids, proteins. Which of these is not characteristic of catabolic pathways? They are not activated by high cellular energy. High cellular energy is talking about high levels of ATP, and catabolic pathways, or your breakdown pathways, are going to be inhibited by ATP. That's in your catabolic anabolic review slides, the last 2 slides of Unit 1. Not found in muscle cells under normal conditions. 2,3-BPG is red blood cells. Anything involved in glycolysis will be found in all your muscles, and remember, creatine is associated with muscles as well. Vitamins regulating calcium and phosphorus metabolism. Let's review what all your vitamins do. Vitamin D is the calcium and phosphorus. Ascorbate is the antioxidant. Vitamin A is cell growth and proliferation. Now for this one, look on slides in Unit 3, slides 9 and 10, list this, and I think there's a typo on there, because when I look through all of the reaction types, what the enzyme classes, I don't see either of these 2. On his answer key, he has this one circled. But when you go through and list all of the slides, enzyme class 1, there's only one redox reaction. That's number 6. There's the most transferase reactions. That's 1, 3, 7, and 10. Those are all kinases. Kinases are transferases. Enzyme class 5, isomerases. That's 2, 5, and 8. And the one not listed here is lyases, enzyme class number 4. And there are 2 lyases. Reaction number 4, aldolase, and number 9, enolase. Based on its functional definition, this molecule does not belong in the group. All of these are going to be coenzyme vitamins. Vitamin D is a steroid hormone. Also, functional definition, you could go with these are all B vitamins. All B vitamins and vitamin C are all your water soluble, and A, D, E, and K, all your lipid soluble. This class of enzyme catalyzes reactions that are not typically regulated. Isomerases are not typically regulated. I think lyases would work for that one as well. Ligases and hydrolases are always regulated, and number 1 and 2 can be regulatory. Which vitamin is particularly important for normal blood function? That is vitamin K. You can review in Unit 1, Slide 53. Some of those vitamins. K is blood coagulation. These molecules is not known as a second messenger. So off the bat, these bottom 3 are all your classical second messengers. Cyclic AMP is another one. IRSP works as a second messenger in the insulin pathway. G protein is more of an amplification role. So G protein activates cyclic AMP, so it's not a second messenger itself. So that's the answer. Oops. All right, number 16, which is directly turned off by dephosphorylation. So remember in Unit 2, go back and review all of your off switches for the pathway. That's going to be the insulin receptor. The insulin receptor is the tyrosine kinase receptors. We skip down to number 19 real quick. Tyrosine kinase, insulin receptor. That's the one that autophosphorylates to turn on, and then is turned off by dephosphorylation. Number 17 is a harder one. It's found on Unit 2, Slide 51. I'm not sure I would have gotten this one here. The answer is glucagon. It's kind of a straight memorization question. For number 18, this reaction, ATP to cyclic AMP. The answer is D, stimulated by fasting. All of these are dealing with some form of GMP, so those are off right off the bat. You learned this in Biochem 1 briefly. It's the cyclooxygenase. That's the enzyme that's inhibited by the NSAIDs. Also, stimulated by fasting. If you're fasting, you're not eating, therefore you don't have a lot of energy, so you're breaking down ATP. So that's the answer there. These signaling molecules is not generated from a membrane lipid. 2 ways to know this one. You can either know epinephrine comes from tyrosine, or you can know all of these other ones have to do with lipids. Diacylglycerol. These have lipid-based, and vitamin D is cholesterol-based. Cholesterol you'll also use synonymously with membrane lipid, but it's not the only membrane lipid, obviously. This membrane-bound protein is a 7T-M. That's your alpha receptor. The beta receptor would work as well. there's one picture. Adenylate cyclase actually has 12 alpha helical domains. This is that tyrosine kinase dimer. Remember, epinephrine is going to bind to a 7-TM receptor, whether it be alpha or beta. So which protein has 3 subunits? That's going to be the G protein, has the alpha, beta, and the gamma. Tyrosine kinase has a regulatory and a catalytic, so 2 total. Insulin receptor has 2 subunits, and then 2 of the things come together to form the full receptor. And pKa has 4 subunits as well. I just mentioned a slide about the adenylate cyclase. If you're looking for that, it's Unit 2, Slide 21. Which small molecule does not appear to be important for vigorous exercise? That's going to be PIP3. That one's generated only with the insulin pathway. So these other ones, vigorous exercise, so you automatically think epinephrine. In the alpha pathway, you're taking PIP2 and converting it to IP3 plus DAG. IP3 then signals the release of calcium, and these 2 things bind to PKC. So all of these metabolites would work, and that gets rid of these 3. Also, we're going to stimulate the beta pathway, which goes from the G protein to adenylate cyclase. It's going to stimulate cyclic AMP, and then eventually pKa. So all of these are stimulated with vigorous exercise as well. Make sure you can rattle those off really quickly. You're definitely going to see a question like this. This is a classical Bracho question, which is or is not a typical response to normal fasting or exercise or normal eating. In this case, normal fasting, you're not going to get an increase in cyclic GMP, you're going to get an increase in cyclic AMP. Everything else works. Just make sure you're familiar with the systemic effects of glucagon, epinephrine, and insulin. Number 25 is another hard one. Straight memorization off of Unit 2, Slide 60. PKC is the one not listed there. Got no tricks for that one. Which protein is found in all cells? GLUT1 and GLUT3 are in all cells. Glucagon is mostly liver. pKa from epinephrine says most cells, but not all. And insulin receptors aren't going to be in all cells either. This metabolite is generated in all cells in significant amounts. So usually what he's going for here is any metabolites of glycolysis, any metabolites or any enzymes. So in this case, 1,3-BPG is going to be in all cells. Acetyl-CoA and FADH2 is in the further oxidation of glucose in the Krebs cycle and oxidative phosphorylation. So these you need a mitochondria for, which red blood cells don't have. Red blood cells are going to go to lactate. Muscle cells can also do that. So you're only really going to find lactate in red blood cells and muscle cells. Fructose 2,6-bisphosphate is going to be in all cells, but in very small amounts. You need a very small amount of it to take effect. Enzyme is not regulated. Physiological conditions. It's going to be this for step number 7. Step number 1 is regulated. Number 10 is regulated, and A and D both correspond to step number 3, which is highly regulated. Make sure you understand step number 3. We're going to see that again here in a second. Which of these molecules is the largest? Enzymes are going to be larger than all the metabolites. So if it's an enzyme, automatically wins. If it's a metabolite, look to see if it has multiple phosphate groups, preferably 2 phosphate groups, like 1,3-BPG or F16 bisphosphate. Those are going to be the larger of the metabolites. And if you were given 1,3-BPG versus F16 bisphosphate together, remember the first set of 5 metabolites all are 6 carbon molecules, and then they break in half and become 3 carbon molecules. So this is going to be bigger if it comes to that. It's a pretty involved question. So moving on to the next one, number 30 here, you're going to find the illustration of liver sequestration, Unit 3, slide 79. Remember, hepatic glucokinase is a glucose sensor. Glucose is the answer. I think on the previous test you said which one stimulates the sequestration instead of release, and that's F6 phosphate. Which molecule does not appear to regulate glycolysis in the heart? It's an easy one. Glucagon is liver only. So in every test, there's pretty much one where you need to know the full reaction for it. This is the one. So you have to know that this is incorrect. You have to switch this with NAD+ here. Everything else about it, about reaction number 6, is correct. So key takeaway: oxidation and phosphorylation. Another tough one here, HIF1 is on slide 57 of Unit 3. Straight memorizing that GLUT2 is not on the list. So there's been, I think, 3 straight memorizing ones. I don't think I would have got any of them. I would have guessed. Which metabolite is not generated in brain cells under normal conditions? Lactate is red blood cells and muscle only. Which enzyme is the most immediate activator for the synthesis of fructose 2,6-bisphosphate? So fructose 2,6-bisphosphate, the most potent activator of PFK1 for step number 3. So it's a trickier question because it's saying the most immediate activator for the synthesis. So who actually synthesizes this metabolite here? That's PFK2. PFK2 2,6-biphosphate. But it doesn't activate the synthesis. PP1 from insulin, that's the most immediate activator of PFK2. A little bit tricky on the wording there. I think on the previous test he said which enzyme is the most immediate activator for the degradation of this molecule. And again, this one actually does the degradation, but pKa would have been the answer because pKa activates the degradation of fructose 2,6-bisphosphate by this enzyme. pKa activates FBase too. So again, review step number 3 of glycolysis. Which metabolite is known to stimulate glycolysis in the muscle and in the heart and to inhibit fatty acid synthesis in the liver? So if we only look at the first part of this problem, it's known to stimulate glycolysis in muscle. And we're looking at this, so it has to do with energy charge. AMP is going to be the answer here. AMP is low energy charge. When you have low energy, glycolysis needs to happen. ATP, high energy, would inhibit it. So the rest is just kind of extra stuff to mess you up. Obviously, if you're stimulating glycolysis in the muscle, the heart is a muscle, so that's extra information. And if you're stimulating glycolysis, if you're trying to make ATP, because you have a low energy charge, you're not exactly having extra energy in order to store as fat. So fatty acid synthesis is going to be impaired as well. Protein does not participate in the rapid release of insulin. Make sure to review slide 68 in Unit 3. LDH has nothing to do with that. Which cells have the lowest overall daily rate of glycolysis in a typical healthy individual? So red blood cells are doing glycolysis for all of their energy, brain cells for all of their energy, so it comes down to the heart and the liver. The answer is liver, and that's because the heart cells are using a combination of fatty acids and glucose all the time, and the liver's job is to, it can use fatty acids and glucose as well, but it's also responsible for making glucose for everything else. So I can't really point to any particular slide on this one. It's trying to understand the concept that if the liver has to make glucose for the rest of the body when there's, you're not eating glucose, then it's going to be using glucose via glycolysis the least amount. It's a trickier one as well. So metabolites not generated by red blood cells. Remember, it goes from pyruvate to lactate. It stops there. Normally, in most cells, it's going to go from pyruvate to acetyl-CoA into the Krebs cycle. For red blood cells, it stops there. So review everything about red blood cells. I think it's come up like 5 times throughout this test. This is glycolysis, red blood cell specific, red blood cell, and muscle cells. This last one, we're talking about inhibiting pyruvate kinase, or enzyme number 10, and that's going to be found on slide 77, unit 3. At the very bottom of it, it says these 4 inhibit the enzyme. Fructose 1,6-bisphosphate is the feed-forward stimulation, which activates it. So that is the answer. Alright, hope this helps. Good luck on your test. I'll see you guys next time.